Good day everyone, we are the Group 9 and we will be reporting about the blood and tissue nematodes. So the lecture part of our parasitology course focused on the different vectors, different hosts, the different signs and symptoms and diseases brought by these nematodes. So here in our report, we'll be reporting and focusing more on the morphology, the microscopic features, and also the different laboratory diagnostic methods of these nematodes. So first we have the W. bancrofti.
Bouturera bancrofti is a parasitic filarial nematode spread by a mosquito vector. It is one of the three parasites that cause lymphatic filariasis. The morphology of Bouturera bancrofti is the most significant differentiation from other species. It has three forms: the microfilariae, the third stage larva, and adult form. The third larva stage is the infective stage of the parasite. The morphology of microfilariae has a size of 240 to 300 micrometer long. A sheath is present. The nuclei is equally distributed. The tip of the tail is free of nuclei and the cephalic space is shorter with a length-to-width ratio of 1:1. The microfilariae or larva stage of Bouturera bancrofti appear as colorless and transparent bodies with blunt heads and pointed tails. It can take several months for the microfilariae to sexually mature, and they can live up to 70 days in human blood. As you can see in the picture, a hyaline sheath is seen at the tail and at the head of the organism. It is a sac-like envelope which is much longer, 359 mm, than the larval body represents the chorionic envelope of the eggs. It remains as investing membrane around the larva. It appears as colorless sheath and gem-sustained. The tail, which is pointed, is free from nuclei. The head, the picture on the right, is blunt, and the space on the anterior end devoid of granules is seen, called as cephalic space. The dark spots or dots seen in the picture are nuclei that appear as granules in the central axis of the body and extend from the head to the tail end, except the terminal 5% of the tip of the tail as illustrated in the previous picture. This is the distinguishing feature of the parasite. The granules are broken at definite places, serving as the landmarks for identification of the species. The morphology of the adult worm, it has the appearance of white color, assumed like a thread-like appearance. The size of the female is 40 to 100 mm. For the male, the size is 20 to 40 mm. Other features for the female is vulva is close to the position of the middle of their esophagus. For the male, it has a finger-like tail appearance. It lacks digestive system, for it obtains its nourishment from the leaf of the lymphatic system of its host. These images show scanning electron microscopy of a Bouturera bancrofti adult worm. Image A, anterior region of nematode body showing a row of swollen bands, long digit form, spherical and spike-like projections. Image B, detail of female tail tip. Image C, anterior region of Bouturera bancrofti with details of cuticular alterations like long digit form, spherical and spike, such as projections. Image D, detail of spike-like projections close to a row of spherical protuberances. Image E, surface of Bouturera bancrofti showing the cuticle with a row of puff-up and deflated structures. Image F, surface of Bouturera bancrofti showing in detail lateral region with cuticle forming a swollen band.
The adult worm has the appearance of long transparent hair-like structure. It is creamy white in color, and as mentioned in the table of the previous slide, the female adult worm is much longer than the male adult worm of the parasite. The both ends of the adult worm are tapering. The male measures 0.1 mm in thickness, and the tail end is curved ventrally and contains two spicules of unequal length. The female are longer than male, measure 0.2 to 0.3 mm in thickness. Its tail end is narrow and abruptly pointed, and the female are oviparous. For the diagnosis of Bouturera bancrofti, its diagnosing active infection is through the identification of microfilariae in a blood smear by microscopic examination. Microfilariae can be detected microscopically in blood smear using blood that was obtained at night 10 p.m. to 2 a.m. The microfilaria periodicity is nocturnal organisms that are present in high number in peripheral blood at night, which is why must be obtained at night, specifically 10 p.m. to 2 a.m. The thick smear should be made and stained by giemsa or hematoxylin and eosin for the microscopic examination. So this is antigen detection, an immunochromatography test for the detection of Wuchereria bruncrusti antigen. Antigen detection, a Lear filariasis test strip, it is point care diagnostic tool that detect circulating filaria antigen in human blood, serum, and plasma. This tool may be useful for assessing the impact of treatment on adult filarial worm in individual and community. So for the antibody detection, serologic enzyme immunoassay tests including antiviral IgG1 and IgG4. So for the polymerase chain reaction or PCR test, the PCR test is done if the individual has a microfilarial infection and to detect microfilariae DNA.
So now I am going to tackle about the Bergia malayi. So the Bergia malayi is a filarial worm that belongs to phylum Nematoda, which is one of the three causative agents of elephantiasis or the lymphatic filariasis in humans. And this has two morphologic forms, the adult worm and the microfilariae or the larvae. So as to the morphology of the microfilariae, it has a size of 200 to 280 micrometer long. It has a sheath present, and the arrangement of the nuclei at the tip of the tail, as what we can see in the image below, is that it has two terminal nuclei which are distinctly separated from each other, and it has a blunt head and a pointed tail. In the next picture, we could observe the head space twice as long as it is broad, and it is usually sheeted, and the sheet stains red with giemsa. So it is closely folded angular, and its body was crowded with nuclei. Its tail is tapers irregularly around two nuclei which are connected by a fine thread. So beside it is a picture that points to the two nucleus at the posterior end, and they are called the subterminal and the terminal nucleus, and they lie folded with the head close to the tail. Another picture that shows that the nuclei is present at the extreme tip of the tail, and the other is in midway between the tip and the posterior column nuclei. So the presence of these two distinct nuclei at the tip of the tail is the distinguishing feature of microfilaria of the Bergia malayi. So as to the adult worm, it is similar to the Wuchereria barncrofti, but are smaller in size. It appears white in color and thread-like in appearance. So the female and the male adult worm differs in its size, in which the male has 53 millimeter, while the female is 53 millimeter, while the male is 24 millimeter, which is shorter. So the adult Bergia malayi are long and slender with smooth cuticle, kinked, and has a long cephalic space having a length to width ratio of 2:1. The head is slightly swollen and has two circles of a well-defined papillae. So the tail of the Bergia malayi is ventrally curved. In the picture shows the male and female Bergia malayi which varies in size, and as what I've said a while ago, the female worm is longer than the male worm. This only shows that there is a sexual dimorphism exists between the adult female and the male. Another picture that shows a male and a female, in which the male coils around the female with his curved area over the female genital pore, and the male uses their spicules to hold the female during the copulation. So beside it is an image that the worm resembles like a delicate white thread. Well, on this image, we could see the lateral section of the Bergia malayi female in a lymph node biopsy, which uses a trichome strain, and a feature typical of filarial nematodes are visible. So the striated appearance of the muscle cell and the nuclei of the uterine and the intestinal cell are evident. And beside it is a closer view of the specimen. There we could see the developing microfilariae within the uteri. So as to the laboratory diagnosis, the diagnostic methods are similar to those of the Wuchereria bancrofti and which includes the thin and thick blood films, wet preparation and concentration, the polymerase chain reaction, ultrasonography, antigen, and antibody detection. So the standard method for diagnosing active infection is the identification of microfilariae in the blood smear by microscopic examination. So one method is the peripheral blood smear, which includes the thick and the thin stain with Shimsa or hematoxylin and eosin, and the subsequent morphologies, and this is for the subsequent morphology species identification. So for increased sensitivity, concentration technique could be utilized. So NOT technique uses 2% formalin, and this is done by centrifugation of blood sample lies in 2% formalin of the filtration through the polycarbonate membrane. Another method is the ultrasonography, in which the infection with the Burgea malayi not only affect the structure and the function of lymphatic vessel, but also it is associated with extralymphatic pathology and the diseases, since it is now possible to detect living adult worm by ultrasonography. A great deal of emphasis is placed on the lymphatic pathology. Next is the antigen detection and antibody detection. So using an immunoassay for circulating filarial antigen constitute a useful diagnostic approach because the sensitivity for detection of microfilariae can be low and variable. So unlike microfilariae with nocturnal periodicity, filarial antigen can be detected in blood sample collected at any time of the day. Currently, this test is not licensed for use in United States and cannot be used for patient diagnosis. On the other hand, antibody detection is a uses serologic enzyme immunoassay test, including the antifilarial IgG1 and IgG4, provide an alternative to microscopic detection of microfilariae for the diagnosis of lymphatic filariasis. So this serologic technique provides an alternative to microscopic detection of microfilariae for the diagnosis of lymphatic filariasis, and patients with active filarial infections typically have elevated levels of antifilarial IgG4 in blood, and these can be detected using the routine assays. So another thing is the specimen biopsy, which is showed in the picture above earlier, in which adults of species can be identified in a lymphatic tissue. Lastly, serum could be utilized. However, current tests lack both sensitivity and specificity, and most people from the region of endemicity will have positive serologic response. So this response may be due to exposure of non-human filarial antigen from infected mosquitoes, and filarial antigen may cross-react with antibodies to other parasitic diseases. So another nematode is to be discussed.
Our next filarial nematode is the Loa loa, or also known as the eyeworm. The Loa loa is a blood-dwelling nematode that resides in the subcutaneous tissues. This type of nematode migrates for a period of time and causes temporary inflammatory reactions such as calabar swelling. The Loa loa is carried mostly by the bite of a Chrysops deerfly species, specifically the Chrysops silasiae and the Chrysops dimidiata, where they are primarily located in West and Central Africa.
So for the morphology of the microfilary of the Loa Loa, it measures at around 248 to 300 micrometers long and is sheathed. Its body nuclei is equally distributed, and the nuclei arrangement exhibits a distinct continuous row that extends up to the tip of its tail. Its cephalic space is shorter, with a length-to-width ratio of 1:1. Its body also has irregular curves and resembles a corkscrew appearance. In this illustration, we can see that the sheath is present and that its nuclei is equally distributed as well as arranged continuously up to the tip of its tail without large spacing. Next, in the first photo, We can observe that the lower lower microfilariae is sheeted and that a short cephalic space is seen on its anterior. On the second photo, we can see that the sheath is quite dull. The sheath of the lower microfilariae does not stain with giemsa and may shed as well as be inconspicuous if there is a delay in the blood processing.
So next is our morphology of the adult worm Loa loa. Like other filarial nematodes, its appearance is thin, whitish, and assumes a cylindrical, thread-like appearance. As for its size, the male is smaller and is typical one-half of the length of the female adult worm, wherein the female is usually 38 to 72 micrometers and the male is 28 to 35 micrometers. Other features include the surface of the body covered with small knobs, the anterior tapers to a narrow head, and the lack of digestive system. Also, the adult face becomes apparent as it enters the conjunctiva of the eye, hence the prevalent term African eyeworm. In our first set of pictures here as shown, these are adult loa loa which were just removed from the eye of a patient. In our second set of pictures here, especially the one that shows the cuticular bosses or also known as knobs are observed here in this picture. Many nematodes have knobs which are scattered over the cuticle and is thought to have a role in the attachment of the worm to the host intestinal wall. The second picture beside it is a close-up picture of an adult loa loa. In our third set of pictures, in this image, here we have an adult female loa loa. Exhibit a whole worm in which we can observe the description that was mentioned before on the appearance of an adult worm loa loa, which is thin, whitish, and assumes a cylindrical thread-like appearance. The letter H or the small h representing its head and the letter T, the tail. In Exhibit B, here we have the anterior part of the adult female loa loa, where the small b is showing the narrow straight mouth of the loa loa and O its muscular esophagus. Last, in our Exhibit C, here we have the posterior part of the adult female loa loa. The small a shows the anus, and the letter U is the posterior uterine loop of the posterior uterus.
Definitive diagnosis of the lower lower, it is done by collecting blood samples for germ staining based on its periodicity, and it exhibits a diurnal periodicity. Microfilariae can be detected by obtaining a blood sample during daytime with its peak transit time of 10 a.m. to 2 p.m. It can also be found in the lungs during its non-circulation phase. Its periodicity coincides with the maximum activity hours of the Chrysops vector. Next, the illustration at the side is the knots technique. It is performed to aid in the diagnosis of loiasis by increasing the likelihood of the procedure in individuals with diminished or reduced number of microfilariae. As what was mentioned in the previous parasite, this technique includes the centrifugation of a blood sample that was sliced in 2% formalin. Thirdly, serological tests can also be performed to detect loa loa, but it cannot determine if the infection is recent or not. Typically, if the blood smear presents a negative result, but the clinical hypothesis of loiasis is high, serological tests can be done in aiming to exclude the infection. Serological tests include the use of lower rapid test, which has a high sensitivity and can be useful for the diagnosis of occult loiasis. ELISA can also be used for serological testing in the detection of loa loa since it is also sensitive, but it only differs in the extent of cross-reactivity with other parasites. With this, ELISA is less useful in endemic contexts as it yields more false positive results compared to LOA antibody rapid test. Lastly, there is also a newly developed technology called the LOA scope, wherein a phone can be turned into a blood parasite detector. Fluoscope is a 3D printed point-of-care testing device that only needs a drop of blood to function. This device aids in determining which treatment or lowiasis medicine will be given to the patient. With this, if the fluoscope finds the individual to have a high microfill amount, dimethylcarbamazine or albendazole may be given and ivermectin if otherwise.
So next, we have the Onchocerca volvulus. And we already know that the Onchocerca volvulus is a filarial nematode which causes river blindness and also is transmitted through the bites of black fly vectors. Since we are already very knowledgeable or familiarized with this aspect of the Onchocerca volvulus, let's focus more on the morphology and also the laboratory diagnosis of this parasite. So let's talk about first the morphology. So the morphology of the Onchocerca volvulus shows that the Onchocerca volvulus undergoes two stages, the larval stage, which is called the microfilaria, and the other stage is the adult worm. So let's talk about first the microfilaria of the Onchocerca volvulus. So the Onchocerca volvulus microfilaria has a size ranging from 150 up to 355 micrometers in length, and it does not contain any sheath in its structure. Also, it contains body nuclei. So this body nuclei pertains to the nuclei distributed throughout the body, and the body nuclei of the Onchocerca volvulus microfilaria is equally distributed. However, it does not extend up to the tip of the tail. So again, the size 150 to 355 micrometers in length, sheath is absent, body nuclei equally distributed, and the arrangement of the nuclei at the tip of the tail does not extend up to the tip of the tail. So here we have the cartoon version or the drawing version of the Onchocerca volvulus microfilaria. And as you can see from the picture, we could see that the body nuclei is distributed throughout the body equally. However, it does not go up to the tip of the tail, or there is no terminal nucleus or terminal nuclei at the tip of the tail. So this terminal nucleus that I'm talking about is the nucleus present at the tip of the tail. However, it is not present in the Onchocerca volvulus microfilaria. Also, the other feature that we could see in this drawing is that the Onchocerca volvulus microfilaria does not contain any sheath. Next, so this we have here the microscopic image of the Onchocerca volvulus microfilaria, and as you can see, again, the body nuclei throughout the body is equally distributed. Also, there's no presence of sheath, and there is no terminal nucleus or terminal nuclei in this image. In here, we have more images of the microfilaria of the Onchocerca volvulus. And as you can see, again, in the middle, we have another drawing version. On the sides, we have the microscopic versions of the microfilaria Onchocerca volvulus. However, I am giving you or I am showing you many images of the Onchocerca volvulus microfilaria since we all know that the microfilaria is not stagnant or does not move. It does move, and it means that what we see under the microscope may not be at a fixed position always since it moves. It can coil, it can curve into any direction. So because of the unfixed position that we may see under the microscope regarding the Onchocerca volvulus microfilaria, we just have to remember the three distinct features of this microfilaria. And again, those three distinct features are the no sheath, no sheath, no terminal nuclei, and then body nuclei that is distributed equally throughout the body. If we are able to remember these distinct
three distinct features of the Onchocerca volvulus microfilaria, it will be easy to detect or identify this parasite, this specific stage of the parasite under the microscope. So next we have the adult worm of the Onchocerca volvulus. So the adult worm of the Onchocerca volvulus has an appearance of somewhat wire-like or very thin appearance, and its size for the female adult worm can have up to can be measuring up to 500 millimeters. And for the male, 25 to 50 millimeters. However, there are sources having varying descriptions of the size of the adult worm on Onchocerca volvulus. However, what is sure in all of the sources is that the female adult worm is always longer than the male adult worm. So for the other features, so both the male and the female adult worm of the Onchocerca volvulus can coil up in knots inside the infective skin nodules. So here we have three images, and these three images are only of the female adult worm, and we were not able to successfully search or retrieve any female male adult worm rather of the Onchocerca volvulus. But what is sure is that both the female and male adult worm have similar structures, except for the presence of the uterine tubes. So here we have the first image. The image here with the arrows is actually a cross section of an adult female Onchocerca volvulus coming from a biopsy of a scalp nodule from a patient in Liberia. So as you can see, we have red, blue, and green arrows, and the red arrows are pointing actually to the female adult worms uterine tubes or the uterus. Um, the blue arrow is pointing at the intestine of the female adult worm, and the green arrows are pointing at the cuticle of the female adult worm. So also another feature to focus in this image is that the uterine tube. So the uterine tubes here in the image does not contain any microfilaria, and this means that the adult female worm here is not gravid yet. But when it becomes gravid, its image will be similar to the middle image in the PowerPoint. So as you can see in the middle image, the uterus is filled with numerous microfilariae, and it also has cuticles, and also the intestine. And as you can see on the third image, the third image is actually also a female adult worm, but it is just the close up image of the uterus containing the microfilariae of the female adult worm. So again, um, we were not able to retrieve pictures or images of the adult worm of the female sex. However, the female and the male adult worms have similar structures except for the presence of the uterine tubes of the females. So that's all for the morphology of the Onchocerca volvulus.
So let us move on to our laboratory diagnosis of Onchocerca volvulus. So the first one is the multiple gemshell stain slides of tissue biopsies. It is through a skin snip, in which it is the most common test used and best detected to diagnose onchocerciasis. This procedure takes small shavings of the skin from different parts of the body, as you can see in the picture, and place them in saline for 24 hours to draw out the larvae. Tissues will be prepared and stained for microscopic examination, and this makes it much easier to spot the parasite through a microscope. But the biggest downside to this method is that it doesn't always catch the disease in people who have a light infection. Next is the serological test. Antibody tests can detect onchocerciasis infection but can't tell the difference between past or current infection. For that reason, they aren't all that helpful in places where the parasite is common, though they can help diagnose the disease in people who have visited such area. Also, this test is fairly rare outside of research setting. Next is the PRC assay. It is it successfully detects low-level infection, and PRC-based assay is significantly more sensitive than current methods and overcomes many deficiencies of parasitologic and serologic methodologies in diagnosing active onchocerciasis. The fourth one is the removing and examining nodules. So, if nodules are under the skin, one or more can be surgically removed to see if adult worms are present. And lastly is the slit lamp examination. This method uses a special microscope and lights like the kinds used during a routine eye exam to see and examine the cornea and anterior chamber of the eye because it is likely where you can mostly see the parasite lurking or the lesion they cause are visible.
So this is the parasite Manzonella otzardi. It is a New World filarial nematode which is noted to have a patchy distribution. It is actually among the three causative agents of the Manzonelliosis disease, which is then commonly transmitted by your culicoides biting midges or your Simulium black flies. Now, moving on to the morphology of your Manzonella otzardi filiaria, it is characterized to be about 220 micrometers long, and they are usually slender, they are clear, and they have a tapered or a button hook. It is noted that these microfiliaria are unsheathted. And their nuclei arrangement actually is such that there are many nuclei. Again, there are many nuclei present in the body of the microfiliariae. However, they do not extend to the tip of the tail. So, as you can observe in this slide, the microfiliariae or the microfilaria, because singular, it is indeed unsheatheded. It is relatively slender. But more importantly, again, there is a fair or a heavy amount of nuclei found in the body, but the tip of the tail is free of nuclei. Again, the tip or the tail, the tip of the tail has no presence of nuclei. The same is observed in this next slide because it shows the features of the microfiliariae in more detail. So again, it has been established that these can be 150 to 200 by 4 micrometers long. It is unsheathed, the tail is long and pointed, and again free of nuclei. And the nuclei, speaking of nuclei, it is fine and it is plentiful and it is well distributed in the body. As you can see, there are segments, there is separation. So moving on to the morphology of the adult worm or the adult form of your Manzanella ozardi is that it appears to be quite bilaterally symmetrical. And it is cylindrical much like the other nematodes. Well, not all nematodes, but I guess for the most part, other nematodes are also symmetrical, they are also cylindrical. So it comes in two sizes, one of that for a male and another for a female variant. Female worms are generally bigger. They are found to be 65 to 81 millimeters long, while the size of the male worms were initially unknown. However, it is accepted that the male worms may reach the length of 24 to 28 millimeters in length, as reflected in a study conducted with patas monkeys, which has resulted in the collection of adult male Manzanella ozarti worms. So again, there was a study conducted with patas monkeys, which resulted in the collection of your adult male the adult form male Manzanella ozarti worms which again had those dimensions. They had those dimensions. Its other features would include the presence of longitudinal muscles which run along the body of the adult worm. Hence, this is also complemented by the presence of dorsal, ventral, and longitudinal nerve cords which are connected to the said muscles. These adult worms are long and slender and they produce thousands of offsprings or microfiliariae at a time. So as seen in these photos or micrographs, it is observed that these worms have little to no asymmetry at all. So what I am trying to say is that they are symmetrical. So again, yes, they are bilaterally symmetrical and they do appear to be long, they are indeed slender, and they are indeed cylindrical. So in terms of diagnosis, the main method used for the diagnosis of Manzanella ozardi is through peripheral blood smears. Now, the reasoning behind this is because that the diagnosis relies on finding microfilariae in the blood. Now, actually, some sources would say that skin snips are also appropriate. However, according to the CDC, Manzanella odzardi is more commonly associated with microfilariae in the blood, while microfilariae in skin snips is more associated to a related parasite known as your Manzanella streptocerca. So the blood can be drawn at any time since Manzanella odzardi displays no periodicity. So again, no periodicity. So what does no periodicity imply? Now, when you say periodicity, it is often associated to a cycle. Again, a cycle or a periodic increase in numbers of the organism in the blood. So perhaps the organism is more active at a certain time of the day. So saying that there is no periodicity connotes that there is no cycle present, there is no interval, and therefore blood can be drawn at any time. And as you may understand, time will not be that much of a variable. If you're going to be collecting or drawing blood in the morning, afternoon, or evening, it will not make much of a difference. So this species actually requires more research and more studies because all that it can do, and how it can be diagnosed has great potential for discovery. Now, why am I saying this? I'm saying this because there has been a study conducted by Viana, Martins et al. And it is interesting because it was about the Manzanella odzardi cases in the Amazon. So what made these cases rather interesting is that corneal lesions were noted in some of those infected with Manzanella odzardi. Specifically, in two patients, a biopsy of the limbal conjunctiva adjacent to the nummular keratitis was carried out and blood collected from the surgical wound for microfilariae investigation was done by thick blood film examinations. Now, before we continue, what is nummular keratitis? So, nummular keratitis is among the features of keratoconjunctivitis wherein there is inflammation and also there is a presence of a certain or a specific infiltrate in the eye. So in the case of Manzanella odzardi, this would imply that there is inflammation, but there is also a presence of your Manzanella odzardi. So uh, what was found in this study or in this research was that there is a positive correlation between corneal biomicroscopic and confocal lesions and your Manzanella odzarti microfilariemia. So why would this be interesting or why would this be unique? Now, because if you are going to be basing this on the life cycle, it could be said or they typically say rather that these worms are to reside in your subcutaneous tissues. The notion of this organism being present or causing damage to the eye is eye-opening. I'm sorry, but yes, it is eye-opening because it is among the few documented cases wherein the organism strayed away from its original route, and it potentially becomes a new means for diagnosis. However, more study is definitely required because if this has not been documented before, we are now knowing this. And so this opens a gateway to more possibilities into looking how we can better diagnose this Manzanella otzardi. So yes, more study is definitely required. That is all. Thank you.
The last nematode that we're going to discuss is the Manzanella persons. So Manzanella persons is a vector-borne human filarial nematode that is transmitted by tiny blood-sucking flies called midges. So in the micrograph presented, we can see the microfilaria of M. persons in a thick blood smear stained with Gemsa. So Gemsa stain microscopic examination is actually essential in order to identify the characteristics microfilariae. So as we can see, its morphological characteristics are quite similar to the Manzanella odzardi. So it is unsheathed and it is approximately 200 micrometer in length. But they can also vary in measurement and in form because they have the ability to elongate and contract. So although M. odzardi and M. persons are similar in their sizes and sheath, when you look at the tail, it is actually quite different. So whereas M. odzardi, as mentioned before, has a tapered tail that is usually bent in a small hook-like shape, Manzanella persons, on the other hand, have blunt tails and their nuclei extend to the tip of the tail. So we can also see in the image that there are many nuclei and that they are equally distributed. So there are also micrographs here of microfilaria of M. persistens in a thin blood smear from the same specimen. In thin blood smear, the nematode is quite darker, hence we could not really see clearly the distribution of the nuclei. However, we could see the head spot that sometimes has a V-shaped appearance. For the adult form of the Manzanella persistens, they live mainly in pleural and peritoneal cavities that are only rarely observed. So for its microscopic features, adults are white and thread-like and have been found to be cylindrical in shape. Males are 35 to 56 millimeter long and 45 to 60 micrometer wide. So while females, as we can see, are bigger, they are 70 to 80 millimeter long and 80 to 120 micrometer wide. So the tail is half a coil in females and a full coil in males.
The laboratory diagnosis for Manzanella persistens can be through microscopy, molecular detection, and serological testing. So first is microscopy. Manzanella persistens is usually diagnosed by finding of microfilariae circulating in the blood through the use of the microscope. In order to do this, we need to prepare a thick peripheral blood smear and a thin peripheral blood smear, which is stained with Giemsa or other appropriate stains that is used for detection and identification of the parasite. So as what Ms. Muggle said, we can identify the microfilariae under the microscope if it has no sheath and the size is about 200 micrometers in length. So this is what Manzanella persistens looks like under the microscope. So the concentration techniques may also increase sensitivity. So examples are NOTS concentration, in which we mix 10 ml of 2% formalin with 1 ml saturated blood in a conical centrifuge tube. Then centrifuge it for 2 minutes. After 2 minutes, we let it dry, then fix it and stain it in a glass slide with Giemsa. Then the polycarbonate membrane filtration. The membrane is treated with a wetting agent or the polyvinyl pyrrolidone or the PVP that permits it to be hydrophilic. Filters are 6 to 11 microns thick, so depending on the pore size. It is also excellent for viewing surface samples with optical or electron microscope. So second is the molecular detection. In research settings, real-time PCR or the RT-PCR assays can be used to detect and quantify Manzanella persistens microfilariae, and loop-mediated isothermal amplification assays or LAMP have been developed for field use. So in here, we need to get a sample, then grind it. After that, we need to isolate the DNA and then add the LAMP master mix. And after that, we need to incubate it and wait for the outcome. So lastly, serological testing. Serological assays helps in finding specific antibodies and antigens which are present in the infection, but there may be possible cross-reactions with other filarial species and other parasitic organisms.
So that's all for the blood and tissue nematodes. Thank you for listening to our report. Good day and God bless.